as you know, you were once one cell. Okay? Sperm met egg, and two became one cell. Okay? And now you are trillions of cells. Okay? So that growth of cells is due to what's called the cell cycle. So it combines both what's called interphase, okay, which is between divisions. So that's G1, S, G2. Okay, and then mitosis, which is the splitting of the cells, one become two. Okay, so for growth of you, also for repair and just producing new cells. So this process is going on as we speak in your body. So let's check it out. All right, so we're going to call this cell cycle regulation. Okay, so here we got a new cell. Okay, so it's just been produced. Okay, so the first phase is interphase, which I said is G1, S, and G2 all together. Okay, so G1 is the first stage. Of course, that's going to cause the cell to grow, so it's really active biochemically, so producing enzymes, new organelles, mitochondria, chloroplast if you're a plant, and just growth of things. So it's a lot of activity. Okay? When you get towards the end of this G1 phase, okay, if the cell doesn't want to replicate anymore, okay, it goes into what's called G0. Okay, so that's kind of a dormant stage. It's kind of hanging out, ready to go. Okay, but for it to kick into the S phase, it has to get by this checkpoint, G1 checkpoint. Okay, so a checkpoint is a spot where it will trigger you to go to the next stage. Okay? So to do that, there's an interaction. There's a group of proteins called Cycline proteins. Okay? They're specific, but they bind to an enzyme called cycline-dependent kinases. If you recall, kinases are enzymes which add phosphates to proteins to make them active. Right? So they phosphorylate proteins, and those proteins will then turn on things or do their work. So in this case, when this cyclin gets a certain level, when it's turned on, okay, when everything is checked out here, it's the right size, got enough nutrients, and it's ready to go into S phase. Okay, this cyclin level will go up. It'll activate this CDK, and that'll produce these proteins, which then do their job and move it into S phase. Okay, so now in S phase here, the most important thing that happens there probably is that DNA actually replicates. Okay, DNA is in the nucleus, it's spread out, it's doing its work, there's a lot of activity, okay, but you can't see it. So at that point, when you can't see it with a light microscope, it's called chromatin. Okay. So chromatin you can't see, but in S phase, that's where DNA replication occurs. So you have 23 pairs of chromosomes or 46 chromosomes. Okay, that becomes 92 at this point. Okay, so that's the key thing in S phase. Okay, then we move into G2, which is prep for the just before the cell division. Okay, so G2, you're going to have the preparation aspect. Chromosomes are going to start to condense. Okay, again, there's another cyclin, CDK enzyme activation. Okay. Then it gets to this G2 checkpoint. So at that checkpoint, the key thing is to make sure that DNA is replicated correctly. Okay, so there's a checkpoint there, and then if that happens, okay, then we can move into mitosis. Okay. So from a type mitosis here, I have PMAT, okay, prophase, metaphase, anaphase, telophase. Okay. So the first stage is probably the longest stage. It's called prophase. See, at this point, that chromatin condenses, so you can actually see them. Okay. And we'll talk about this in a PowerPoint, but now you have replicated DNA. They're held together at a point. So when you look at them, it looks, you have a chromosome here, and it's got an exact replica here. And they come together here. Okay, so they're still connected. That's called the central mirror, where they come together. Okay, so they're two identical strands, and now you call them chromatids. Okay, so they're exact copies, so they actually call those sister chromatids. Okay, and they shorten and condense. Okay, then the nuclear envelope starts to disappear during prophase, and these spindle fibers start to radiate out from what are called the centrioles. Okay, centrioles move the pole, and you get these fibers, and these chromatids are now visible with light microscope. Okay. Then at metaphase, they line up at the equator. And we'll go over this in the PowerPoint. Line up on the equator, and they're getting ready. So what you have then is, it almost looks like a cell without a nucleus, and then at the ends you have these centrioles, and then these fibers will radiate out. It kind of looks like a football, okay, kind of oval shaped like that. Okay. And during metaphase, these chromatids line up on the equator. Right? And then the last checkpoint, M checkpoint, is actually between M and A, between metaphase and anaphase. Okay, so it makes sure that the chromatids are all lined up. They're lined up on the middle. Okay, so they're ready to go into anaphase. During anaphase, the chromatids split apart, so they're no longer connected to that centromere. Okay, so they're brought apart. So now you end up with, at the end of anaphase, one set of chromatids is here, one set is over here. Okay, so if you recall, if you're talking about humans, okay, we had 46 chromosomes here. To start, at the end we have 92. Okay, so at this point we have 92 of these chromatids. Okay, but at this point they split apart, so now we have 46 here and 46 here. Okay, so they're exact copies of each other. So this cell is just like this cell, so they're exact copies of each other. Okay, that's in anaphase and telophase are at the ends. The nuclear envelope starts to form around them. Okay, the spindle fibers disappear. Centrioles kind of disappear also. So then the last stage is what is called cytokinesis. In cytokinesis, the cell splits. So cyto means cell, kinesis means to split apart. Okay, so it splits apart, and now you have two cells produced, which are the exact copy of each other. All right. So now, of course, there can be some problems here. One, if the DNA material starts to break down as you age, or you have some sort of mutation, then that can cause the cell not to divide properly, and it causes apoptosis, which means just cell death, so they can't go on. And sometimes these checkpoints don't work, okay, so it should stop, okay, at this point right here, and for some reason the cell just may keep on growing, okay, so nothing's turning it off, so it might be some sort of mutation or something happens, so you have uncontrolled cell growth, and that's not good either. That's actually probably what's responsible for cancer. Well, it is responsible for cancer. Cancer is uncontrolled cell growth, so that's the cell cycle, and it's